Phenol red broth is commonly used to measure bacterial fermentation. In this video, we'll start with the fermentation of two common sugars, glucose and lactose. The fermentation of glucose starts with glycolysis, which results in the production of the energy intermediates NADH and ATP, as well as pyruvate. In fermentation, bacteria utilize pyruvate and its derivatives as alternative electron acceptors to oxygen. The benefit for the cell is to oxidize NADH and regenerate NAD+, which in turn allows for the process of glycolysis to continue. Common byproducts of this process include a variety of different acids and in some cases gases. The fermentation of lactose starts with the hydrolysis of the disaccharide into glucose and galactose. The resulting monosaccharides are further catabolized through glycolysis and fermentation, producing various acids and gases along the way. One medium used to visualize fermentation is phenol red broth. Phenol red broth contains a fermentable carbohydrate and the pH indicator phenol red, which responds to fluctuations in pH by changing color. If a bacterium ferments the provided sugar, the color transitions from red to yellow, indicating that acid was produced as a byproduct of fermentation. Phenol red broth may also contain a small inverted tube called a Durham tube. The purpose of this tube is to capture any carbon dioxide gas generated during fermentation. The loss of liquid volume in this tube, often described as a bubble, indicates that gas was produced as well as acid during the process of fermentation. Now that we've learned about the underlying theory, let's move on to expected results. As we review the results of phenol red, pay attention to two things: the color of the tube, as well as the presence of a bubble. Here is the uninoculated control. The neutral pH of the phenol red appears as a reddish-orange color. Note the appearance of the Durham tube, which is completely filled with liquid. As we transition to the next result, notice how the color jumps out. The yellow coloration, due to a shift in pH towards acidic, indicates the bacterium fermented the supplied sugar with acid end products. The presence of a large bubble in the Durham tube demonstrates gas end products as well. The proper interpretation of this result is fermentation with acid and gas, or AG. Like the previous tube, the yellow coloration indicates the bacterium fermented sugar. Notice, however, the Durham tube. The lack of bubble indicates that this is fermentation with acid-only end products, or A. The next result is in stark contrast to the previous. The red coloration indicates the pH remained neutral due to a lack of fermentation. And because the bacteria was unable to ferment the sugar, notice the lack of gas as well. The proper interpretation for this result is no fermentation, or N. The magenta coloration of the last tube indicates the bacterium catabolized protein, producing the base ammonia, which resulted in a shift towards alkaline conditions. The proper interpretation of this result is no fermentation, but it's labeled as K due to alkaline pH. Phenol red broth is a convenient way to separate members of the gram-negative enteric family, which often differ based on their ability to ferment lactose. To learn more about how proper diaper changing practices are used to avoid infectious outbreaks by enteric bacteria, make sure to read the Focusing on Health section titled Daycare Danger.